Dr. Georgia Titcomb talk for us today. Georgia is an assistant professor of wildlife disease ecology in the Fish and Wildlife and Conservation Biology Department at CSU in the Warner College of Natural Sciences. Resources. Thank you, Alonzo. See?
I didn't even catch that.
And her research spans a wide variety of projects across disease systems based in the U.S. and also sub-Saharan Africa. And her projects are united by the lab's central theme, investigating how humans are impacting wildlife disease and understanding how hotspots affect disease transmission, and then harnessing new tools and techniques to improve our ability to measure and understand diseases. And so today she's going to be talking about some of the work she's been doing in Madagascar. Welcome, Georgia.
All right. Hello, and thank you very much for the invitation to speak today. As Tracy mentioned, I'm going to talk a little bit about some of the projects that I'm involved with based in Madagascar. So in many areas of the world, humans, livestock, and wildlife all live in constant contact. But really, which contacts matter for disease transmission is a central question, and which of those contacts actually matter for different diseases across contexts and landscapes. So the area of the world that I'm going to be talking to you about today is based in the Sava region of northern Madagascar. This region is really well known for its major role in vanilla production, but it also includes the beautiful Marojejy National Park. I realize I need to start my laser pointer here. Beautiful Marojejy National Park, set within this landscape of lush vegetation and exceptionally rich biodiversity. Across this region, there's a striking mosaic of habitats that sit right alongside a lot of different small communities all along this road here. And in these communities, there are lots of interactions among humans, livestock, and wildlife. So the park is home to a remarkably rich array of animals and flora. Madagascar, in fact, has one of the highest levels of endemism in the world. Approximately 80% of all species that are found there can only be found in Madagascar. This includes iconic species like the silky shavak here, which I did get to see when I went out there. My photographs are absolutely terrible, so you're not featuring them here. Including many native birds, reptiles, amphibians, and of course, small mammals, which I'll talk a little bit more about today, including animals like this tenrec, which is actually more closely related to elephants than rodents. Okay. So for this project, we focus on 3 villages that are located outside of the national park. So you can see the outline of the national park here, and then our 3 villages here. Each village has roughly 1,000 to 3,000 inhabitants. And the surrounding landscape is a patchwork of different land uses, as you can see here, rice fields, and then also agroforestry areas around the edges of the national park seen in the mountain areas. And together, these create a highly heterogeneous landscape, all alongside that national park. We can see a pretty stark contrast between the native landscape and the areas where humans are living. Okay, and so within this landscape, one of the main agricultural activities is rice farming and harvesting, as shown in the top 2 pictures up here. There are really sort of different seasons across the year where people grow rice and then move it into the village. So there's a really big transition of resources across different seasons. And then, as mentioned before, vanilla production is a major activity in this region. And again, most of the world's vanilla is coming from this very specific area in Madagascar. And so taken together, really this means that people are living in really close contact with their environments, much like small-scale farmers across the world. And of course, that intimate relationship with the land ends up putting people in contact with livestock and wildlife and the environment quite a lot. And of course, in this area, there's a lot of pathogens and parasites that these people are having to grapple with. That's like useless. Okay, So this project, really a collection of projects, comes from a really large body of collaborative work. So, what I'm showing you today is sort of nested within a much bigger project, all of which is kind of spearheaded by Dr. Charlie Nunn and Dr. Vangi Swaritmalala, based at Association Bahatra and Duke University. And all of these folks are all PIs on various projects, and there's sort of a network of folks working across the world on basically One Health projects based in Madagascar. And so, what I'm going to talk to you about today is a particular project or group of projects that are headed up by several of these different PIs with specific goals. Firstly, of building transmission potential networks that can represent some hypothesized routes of infection. So understanding how people, animals, and the environment are all connected in ways that are relevant to transmission. We also want to identify specific socio-demographic covariates of that risk. So how is that infection unevenly distributed across people? And we want to assess whether those networks can represent hypothesized infection routes and therefore correspond to patterns of infection. Okay, so a few methods, and so this is going to be a little bit different in that I'm going to give you an overview of all of the methods that we kind of employ across lots of the projects up front, and then all of the different projects that I talk about afterwards are going to refer to these. So first of all, a lot of our data come from surveys, whereby in each of the 3 villages, our survey team, so our anthropologist team, again representing our many different disciplines across a One Health sort of collaboration here. So our demographic data was collected by our social network team, basically, and so they went and surveyed inhabitants of each of the villages on their basic demographic characteristics. They also asked questions about their social network. So questions like, who do you go to for help if you need it? Who would come to you for help? Who are your friends? And then we would go and ask those folks similar questions to sort of get a better understanding of the overall village characteristics. And then folks were also asked questions about their interactions with animals and also their knowledge of zoonotic diseases. So trying to understand how people were coming into contact with animals in their environments. Participants were also invited to participate in a GPS sort of movement study here. So folks were compensated with Chrome credits across all of these, actually. And they could opt in to carrying around a GPS logger to show us where they moved on the landscape, and they would sort of renew that on a weekly basis. The number of weeks that people participated was between 1 and 6 weeks, usually about three was about the average. And then we would overlay folks' movement, their utilization distributions, which I'll talk about in a little bit, with the different land classifications to understand how people were intersecting with different parts of the landscape. And then we were also able to even get GPS loggers on some of the domesticated animals, so there's a little bit of data on that as well. People could also opt into providing biological samples, so they could be screened for various parasite infections and pathogen infections, so obviously I'll talk a lot about that. And for this particular presentation, I'm going to be talking about our parasite identification done via metabarcoding, specifically in stool samples. And then at the same time, this could be a whole other project, but our team at Association Bahatra in Madagascar spearheaded this work on small mammal collection and trapping along land use gradients. And there were 7 sort of sites that came from each village in terms of varying levels of habitat degradation. And 3 seasons in each of those villages, and each of the animals that were caught, we collected biological samples for screening. So we found a lot of different pathogens and parasites. So a very brief overview of sort of everything that's been surveyed so far. So Pablo Tortosa out at La Reunion Island, he has done a lot of screening on microparasites. So hantavirus, I'll note that this is not the same hantavirus as the hantavirus that we find here in North America. This is an old world hantavirus, which is Still not great, but a little bit less of a concern. But we did see much higher prevalence in non-native versus native species. There were paramyxoviruses, astroviruses, and leptospira, all of which tended to be higher in non-native species than native species, save for paramyxoviruses. We also found a diverse array of protozoa. The most common were Blastocystis, Cryptosporidium, and Amoeba, and then a really wide diversity of sort of unknown types in small mammals. Lots of helminth diversity as well. So Necator americanus is the human hookworm. I'll talk more about that today, as well as a zoonotic hookworm and other roundworms, and lots of unknown parasites in small mammals. And then work headed up by Casey Ring at UC Santa Barbara. She looked at ectoparasites, trying to understand this diversity of different ticks, fleas, and mites, and she found an interesting partitioning in fleas and ticks in village areas and areas outside of the village, including several tick-borne pathogens as well. But today, I'm going to focus a little bit more on these in the center. So this is our lab at CSU, has really been working more on these gastrointestinal parasites. And so I think this is a really good system for a One Health talk, because GI parasites are highly relevant to One Health and transmission networks. They connect humans, animals, and the environment via water, food systems, waste management, and soil. So we're really looking at like many different layers of the environment here. Parasites are also detectable in non-invasive samples that are relevant to transmission. So a lot of fecal oral parasites are much more easily sampled than, say, bloodborne parasites. And then when you sample it from the environment, that is relevant to the ongoing transmission. So we can make, I think, a few more conclusions about transmission when we're able to detect parasites in those samples. GI parasites are also really interesting because they span this gradient of generalism to specialism, so we can ask and answer a lot of really interesting disease ecology questions about who gets infected relative to maybe a lot of exposure. Is there some filtering based on the degree to which hosts are related to one another? And then GI parasites are also really interesting now, or a lot more tractable system to be looking at, because we now have much better methods to detect them, specifically through metabarcoding, which is what I'm going to talk to you about today, which allows us to now sort of look at very sort of disparately related parasites, whereas previously, we would be amplifying the DNA of all eukaryotes, for example, when we're trying to detect parasites. Okay, so today I'm going to talk to you about 3 sort of One Health stories, and I'm borrowing the One Health Institute's logo here to illustrate, how we've linked humans, animals, and the environment in all of these different case studies here. Okay. So our first story is about Blastocystis. So an estimated 1 billion people worldwide are infected with Blastocystis. It is one of the most common parasites in the world. But Blastocystis is a little bit of a One Health enigma. It's a protozoan parasite that's typically transmitted through fecal-oral pathways, as illustrated here, making it really relevant, again, to those kinds of human-animal environment networks that I mentioned. And one of the major open questions is whether Blastocystis is truly zoonotic, or is actually even a parasite. And if so, under what circumstances does transmission occur? And part of that uncertainty comes from the fact that there are multiple different subtypes of Blastocystis that may vary in their host specificity. And then some of those subtypes are known to infect various wildlife and domestic animals, as well as humans. And so we're really trying, I think now the literature is really trying to understand which subtypes map to which hosts. I'll also note that Blastocystis, while it's really common in a lot of developing countries around the world, it's still present in developed countries, just at lower prevalence. So it's really globally relevant. And so historically, blastocystis was often thought to be closely related to irritable bowel disease and other gastrointestinal problems. But over time, that picture has become a lot more complicated. And there's been a lot of discussion about the variation in pathogenicity depending on the different subtypes. And this has sort of come out of the fact that we found that a lot of people have blastocystis, but they may be asymptomatic, while others also have blastocystis and really suffer the consequences there. And additional papers more recently have come out really trying to link things like diet and microbiome to blastocystis infection. So there's really additional layers and dimensions to this parasite, or commensal, who knows, sort of globally. And I'll point out this paper that actually came out pretty recently that was linking it to healthier diets and even favorable cardiometabolic outcomes. So really, the picture is very complex. But the one takeaway that we can have is that subtypes really matter, okay? So it really is gonna depend on what the subtype is. And historically, we haven't really been able to detect or really spend a lot of energy trying to differentiate among the different subtypes when we sample populations. So in our study system, blastocystis in Madagascar, we found a high prevalence across our villages, about 60 to 70%. This is high. It's not outrageously high, but it is on the higher end of what we might expect. And this is our sample size across our villages. So we're working with roughly 800 people here who have both movement and infection data and socio-demographic data, as I mentioned before. And so we're, again, really interested in figuring out what explains those patterns of blastocystis infection. And so two graduate students from Duke University who I worked with, they really led the analyses on blastocystis in particular. And so Alma Solis, she wrote a paper looking at the socio-demographic covariates, so just looking at how that infection was distributed across different characteristics. And then Tyler Barrett took a network approach where he compared 3 different networks. A zoonotic network, so the degree to which people interacted with animals and shared subtypes. A social network based on sort of friendship, and a household network to see if any of those networks corresponded to infection patterns. So first of all, I'm just going to show you the subtypes, the subtype distribution across hosts here. People typically did not share subtypes with other animals. So really interestingly, what you can see here is people tended to have subtype 1, 2, and 3, shown in yellow, that's humans here. But subtype 3 was very rarely found in other animals, so this was really uncommon. Meanwhile, other animals were infected with blasto. Most notably pigs were the ones that had the highest prevalence. But the takeaway here is that other subtypes are abundant in the system, but people are not getting them. So again, subtype 5 tended to map to pigs, subtype 10 and 14 to cows, and subtype 4 to rats, while humans were really 1, 2, and 3. So that partitioning among our host species is really important, and actually runs a little bit counter to what the literature has implied about blastocystis being more of a zoonotic parasite. Okay, so moving into the socio-demographic relationships, we also found this subtype mattered here. So we found that women were actually more likely than men to be infected by subtype 3. And we found an effect for hand washing in that folks who use soap tended to have lower odds of subtype 1 and 2 infection. Those are the only things that made any sense or came out of our analysis. There was no relationship with wealth, education, age, or animal contacts. And I guess the animal contact pattern makes sense, right, based on the fact that subtypes didn't map across multiple host species. Okay, so we've ruled out several possible things that could be related to blastocystis. But now we want to look at how maybe more complicated relationships among people might explain patterns of infection. So again, Tyler led this work here. Oh, I'm sorry. I should mention here, right? Because because we didn't see any shared subtypes across animals and humans, we could pretty much rule out a zoonotic network because animals just didn't have the same subtypes that humans did. Okay, so a big takeaway from Tyler's work here is that neighbors tend to share infection status rather than friends. So the proportion of close neighbors who were infected was the only network that explained infections, which kind of surprised us. We thought that maybe a social network would be important here, but it wasn't. And this was true across all 3 villages and all subtypes, also kind of surprisingly. And we'll say that the spatial relevance did vary by village to some extent, kind of depending, what the radius is of what you call a neighbor. There was a little bit of that, but generally the folks who were closest to you, that was where our effects were strongest. And so what I'm showing here, oh, you can't actually see, sorry. Okay, so what I'm showing here is a network of folks in one village, and each node is mapped to where that person lives. Nodes are colored based on whether they're infected or not. The edges that connect the nodes is the friendship network. So the first takeaway here is that friends are not neighbors, or neighbors are not friends necessarily. Those networks had no correlation, which also surprised us. But there was definitely a geographic relationship. And you can't see it as well here, but I actually have a little heat map in the background, and there's a river that runs along the side of the village here, and the relative risk ratio near the river is about 3 times higher than the other areas. So there's a pretty clear sort of geographic component, at least we think, happening here. So that's just one example. And so this points, again, or takeaway, to human-human transmission happening, and that neighbors tend to share infection status, but friends don't. And this also points to this possible role of food and water sharing that's geographically dependent. This implies, again, zoonotic dynamic that, originally we might have thought was important, actually isn't. And we should focus on potential shared water sources for any kind of management or intervention. So, zooming out here, the main takeaway is that not all hypothesized networks matter, but taking this holistic kind of approach allowed us to rule out various pathways and identify a sort of pathway that's most relevant to transmission and also intervention. And then our next steps are obviously to look at the water, to test the water and see what might actually be there. Can we detect the subtypes, and are subtypes distributed differently in water sources across the village? And so leading that work is a PhD student in my lab, Christina Blanca, who's here. And she, in collaboration with my master's student, Ali Mujica, is also they're going to go out to Madagascar and sample the water sources, thanks to a grant from the One Health Institute, so thank you. Okay, so 3 One Health stories returning here. We've talked about blastocystis, and now I'm going to talk about hookworms. Okay, so like blastocystis, hookworms are also incredibly common across the world. And an estimated 500 million people worldwide are infected with hookworms, so another really globally important parasite group. Unlike blastocystis, however, there's a lot less debate about how important hookworms are in terms of people's health and whether they sort of really matter for health. They can cause significant morbidity, so while they usually don't cause mortality, in terms of disability-adjusted life years, they are a major cause of sort of problems across the world. And it leads to population-level effects in productivity and health as well. And so one really interesting thing about hookworms is sort of their life cycle that's slightly different from blastocystis, and I'll just briefly mention this here because it's relevant to transmission, right? So hookworm eggs are passed in feces, and those eggs develop in the environment and turn into larvae. Those larvae then move onto vegetation or other areas of the landscape, or plants usually, where they will come into contact with people's skin, usually hands or feet, in a lot of areas of the world where people are not wearing shoes, that's a major transmission route, but also if you're sort of working in agriculture or handling the earth a lot, that's also a potential transmission route. Those larvae will then enter the bloodstream, so they burrow into the skin, enter the bloodstream, enter the circulatory system, go to the lungs where they are then coughed up and then swallowed with the sputum and enter the gastrointestinal tract, and the whole life cycle is repeated again. So honestly, kind of amazing how this evolved in the first place, but important to think about when we think about transmission, because contact with the environment, and not just eating, but physical contact with spaces in the environment, is a likely transmission route. And so that's important for us to remember. So again, environmental transmission, that's proportional to time spent or energy spent in different areas of the landscape. Hookworms are also known, or they're associated with poverty traps, whereby people who have hookworm infections tend to have lower socioeconomic status or fewer resources. And there's this idea of this constant cycle whereby hookworm infections limit people's productivity or ability to also earn additional money, creating this kind of vicious cycle. So there's kind of a long-standing hypothesis that that's happening. So in Madagascar, we found a high prevalence of hookworms, about 50% for the human hookworm, Necator americanus, which I am showing here in red. And then we also found a zoonotic hookworm, Ancylostoma ceylonicum, shown in yellow here, across the villages. We had sort of some variation, but sort of similar general patterns, including several co-infections, as shown here. And I'll note that that zoonotic hookworm was at high prevalence in the dogs that we screened. So it's about 75% of the dogs had this hookworm. So again, we asked what socio-demographic variables predict infection and co-infection. And again, can transmission potential networks describe those patterns of hookworm infection, like we asked with blasto? What we considered, we asked about sex, age, schooling, wealth, and animal contact to see if any of those tracked with infection patterns. And then we also asked about how different networks may map to infection patterns. And so the way that we did that, so this is slightly different from the blastocystis method in terms of our environmental overlap in different land cover types. So we took, as mentioned before, the utilization distribution of people's movements across the landscape and overlaid that with the habitat classifications across each of those villages. And so one individual might look like this, where that one individual is connected to each of the different grid cells in varying strengths depending on how much time they spend there. We then take that and basically repeat that for all of the people in a village. So we have this big bipartite network of people and grid cells linking all of them together. We then do a unipartite projection. So basically then we take each individual, shown by these nodes, and they're connected to each other if they share a grid cell. So if they spend time in the same grid cell. Okay. Oh, and I'll just mention that we'll then make different networks depending on the land cover type of each of those grid cells. So we can have a forest network, we can have a rice field network, a village network, etc. Okay, so moving back to our socio-demographic covariates, what we found. So what I'm showing here is the logogs of infection across each of our variables. Anything you see on the left-hand side of 0 means that there's a negative association. Anything on the right means there's a positive one. And so what we found is that males tended to be more likely to be infected. So that was pretty significant in terms of like across Necator americanus for sure. And then we also found it for co-infections and the zoonotic parasite. We found a potentially significant association with the school level for the zoonotic hookworm. But we did see this poverty trap hypothesis play out here, whereby folks who own fewer material goods, we had to use principal components for that, but generally lower material wealth was associated with higher odds of infection. I guess I explained that in the opposite direction. The more material wealth, the lower your infection odds is really what that's showing you. And then dog ownership was the only other variable that popped out. So and this was, again, only for the zoonotic parasite. So that tracked with our hypotheses as well. Okay, so then moving on to movement and the ways that people use the landscape. We did see that there was an association between the amount of time that people spent in forest fragments and infection odds. And we found that that played out across, whether folks were male or female. And so then that led us to ask, well, does a network of shared forest fragment use predict infection? Okay, so to test this hypothesis, we used what's called a network permutation test, which is the same general approach that we use for the blastocystis analysis. But I'm going to walk us through it a little bit more explicitly here. And the goal, again, of this test is to ask whether a given contact network is associated with transmission patterns, that more so than we would expect by chance. So in this case, let's start with a network, like that network that I showed you earlier, different line covers, where we have nodes that connect people based on whether they share areas of the environment. Nodes are then colored based on their infection status. So our statistic that we calculate is the mean number of cases, K, within one step of an infected case. So in this example, I might take my data and calculate the mean of every single infected case connected to an infected case. So in this example, there's 20 is my mean. Then I randomly reallocate cases across the network. So I take that network and then I randomly distribute cases across that many, many, many times, 1,000 or more times, and calculate that same mean number of cases that are connected to an infected case. So in this example, when I do that, each infected node is connected to 10 other infected nodes. When I do that, I get a distribution of values here. And then I compare my observed K to this distribution. So in this example, I have a pretty significant, doubling in terms of the level of aggregation of infected contacts on that network. Okay, so this was a hypothetical case. What do we see in our data? So our network of secondary forest use corresponded to human hookworm infections only. So I'll talk a little bit more about the zoonotic one soon. But the human infections, in Village M, 23% more connections among infected people than we expect by chance. In Village A, this was a lot more obvious, 47% more. So we're definitely seeing some kind of signal of people's patterns of movement outside the village being associated with infections. Okay, so that was forest fragments, but what about other areas of the landscape? We saw a signal that people tended to use more forest areas, and that was associated with infections, but what about sort of those other areas? So here, there's a slight modification to what I'm showing you here in that my y-axis is now my increase in network strength, and basically what that is I'm just accounting for the amount of time that people spend. So I'm now using a weighted network that accounts for sort of the volume of time that people share rather than just do they share or not, which is a binary network. So same idea, just adding in that accounting for that extra weight. So when I do that, and again across all of these different networks, so we build one for all areas outside the village, for secondary forest, for brushy regrowth, rice fields, and village areas, as well as a house network, which is based on distance of different houses to each other. I can see that in Village A, basically all areas outside, the patterns that people, shared landscape areas outside the village are all significant here, some of them much more so than others, and especially in our secondary forest fragments. Note that areas inside the village and houses have nothing to do with the patterns of infection, which is totally opposite to blastocystis, right? So two parasites doing very different things. We see similar patterns in Village M, so completely, really, really similar things happening here as well. So again, all the areas outside the village are important here. So you may be wondering, well, Georgia, there were 3 villages that you told me about at the beginning. Why are you only showing me 2 here? Well, that's because when we look at the 3rd village, everything falls apart. And we were looking at this and we were like, wow, that's really interesting. This makes a lot of sense. This does not make very much sense. Okay, so none of these were significant, and the house network, there was a little signal there, but honestly the effect size is pretty small, even though it was significant. So what is going on with Village S? To answer this question, we look at the habitat distribution across these different villages, and when we do so, we can see that Village S is very different from the other villages. Specifically, Village S is surrounded by open rice fields and bare ground. And villages A and M are part of this more complex network of forest fragments and brushy regrowth areas with different patches of rice farming, at considerable distances away from people's homes and possibly latrines, right? And so I guess a big takeaway, a key takeaway here is that all of these villages, people in Village S, are operating on a completely different environmental context to people who are living in villages A and M, and that likely has a lot of implications for their daily movements and hygiene patterns, and sort of what's available in terms of privacy for open defecation, which is practiced in all of these locations. So we think that that is almost certainly what's going on here, or at least our evidence seems to point to that. Okay, so I talked about the human hookworm just now. I'm now going to talk about the zoonotic hookworm, which again, I'll remind you, is high prevalence in dogs. And so our results here is basically the same setup as before, but now I'm showing you the zoonotic hookworm, and it clearly depends on the village in terms of whether there are any patterns with the network. So again, Village S, nothing going on there that makes really any sense, or we're not able to adequately explain it. Village M, okay, maybe there's a little bit going on in our brushy regrowth areas, and then a much stronger signal in Village A. Okay, so why is this happening? I'll note that in Village A, it's slightly different from the other two, in that there are 40% fewer dogs than the other locations, and also there's lower prevalence in people. So why on earth are we getting a clearer signal of human movement in an area where there are fewer dogs? So we think what's probably happening is that there may be proportionately more human-human transmission in Village A that we're able to pick up on. The other villages, Village M and S, have proportionately more dogs, especially Village S. So this actually kind of tracks the general number of dogs that are in the area. And so what we think is happening is that the animal reservoir is creating more noise in the system that we simply are not capturing because we've not integrated them into our networks. And while we did put GPS trackers on some dogs, we had about 5 in each village. So we do not have the sufficient sample size to be able to integrate them into these networks and actually test that hypothesis. But if we could, that's exactly what we would do. We would build stronger networks that also include dogs for the zoonotic parasite. And again, I just think that underscores the importance of making sure when you're looking at transmission to make sure you're tracking the movements of all the relevant hosts. And also that it doesn't necessarily, or it may differ depending on the context, which village you're in. Okay, so parasite species matters. That's our takeaway here. We got very different results based on the parasite, even those that can seem similar. And one thing I'll just close with here on this story is that hookworm infection is also tied to cardiovascular health. So we see the parasite species mattering in terms of health and not just transmission. And so this work is ongoing, like right now. Lev Kalinsky is another graduate student who's working with us from Duke University, and he's detected important species level differences in that Necator americanus, that's the human-associated hookworm, is associated with lower BMI and decreased blood pressure, while that zoonotic hookworm has an opposite effect. So big takeaways in terms of context really matters and species really matters. So our big take-home messages here: demographic patterns of infection are also explained by demographic differences in exposure and movement. And that similar parasites, or at least seemingly similar, can have very different transmission patterns and also health effects. So it really depends. And that the most relevant transmission route really depends on that geography and context of each village. Okay, so now I'll move on to my last One Health story here, which is about wildlife parasites. And so this is for my wildlife enthusiasts and conservation biologists who may be present. Okay, so approximately 5 to 15% of vertebrate helminth diversity has been described. Helminths, worms, in other words. And so we're still missing the vast majority of this diversity. And this is really not a small gap, okay? So this likely represents thousands of undescribed parasite species. In fact, estimates suggest that there's roughly twice as many worm parasites of vertebrates as there are actually vertebrate species. There are not twice as many parasite ecologists, so I'm running a pitch here for people to join the team. But this is a big slice of biodiversity that we're missing. And again, as I mentioned, Madagascar has the highest level of endemism in the world. A recent paper, I guess 2020, I don't know if that's recent anymore, but a paper that came out in 2020 mapped the distribution of our knowledge gap across the world here. And I'll just point out that Madagascar here is, verging on nearly 100% of the parasite species that we think are there are like unknown to science. And this has even led the same researchers, actually a big group of parasite ecologists, to come together to propose, thinking a little bit more holistically about parasite diversity, and to even, goodness forbid, think about conserving parasites, or at least understanding their diversity and their patterns across the landscape. So our questions out in Madagascar, in this incredibly biodiverse, rich area of the world, is how are those parasite communities changing in relation to human activities? If you remember that map where there's satellite images that I showed at the beginning, that stark contrast between the national forest and then the areas where folks are living. Are we seeing invasive species bringing new parasites as well? So we also have a good number of invasive species here. And again, as I mentioned, our collaborators at Association Vahatra in Madagascar are really, the amazing naturalists who are spearheading this work. And so it's a really awesome to get to work with them, and I'm highlighting the enormous amount of work that they've done through the pandemic even to continue working on these small mammal trapping projects. Okay, so as I mentioned, our trapping grids span a habitat gradient. So these are Google satellite imagery of, say, village areas, rice fields, brushy regrowth areas, secondary forests, and then intact, semi-intact national forests. And across this habitat gradient where the trap grids are set, we find quite a few invasive species that tend to be associated with village areas, namely black rat, house mouse, and Asian house shrew. But then we also find a really cool array of extraordinary Malagasy small mammals, and I call them SMAMMALs, so excuse me if I say that, which there are a few native rodents, but there's also an enormous diversity of tenrecs, which, as I mentioned, are a completely different group of animals based in Afrotheria, which are very, very different from the rodents that we know. And so, sadly, what we found is that small mammal communities are dominated by invasive species. So we trapped and screened close to 2,000 animals across those 3 villages, and almost half, it might be more than half, were Rattus rattus. There are a lot of rats out there, especially around the rice fields, as you can probably imagine, and in village areas. But it's not all bad news. We also found a substantial number of microgale, which is a native, really small tenrec and incredibly cute. That's sort of our dominant native species. And so today, I'm just going to focus on the parasites that we find in these two and contrast them to each other. And a fun image, this is a a little bit more in proportion with their actual sizes. There are some huge rats out there. Okay, so here I am showing, we're asking how are these parasites changing along a gradient of human activity. So I'm showing here our results for rats in particular. And our first parasite is a common rat coccidian parasite, and looking at it in terms of distance from the village and prevalence on the y-axis. So what we see is a pretty kind of clear decrease in terms of parasite prevalence being higher in rats closer to human habitats. We don't think that humans get rat coccidians. Coccidians tend to be fairly host specific, but just generally observing this pattern here. And we see this consistently across our 3 villages. There's also another protozoan parasite, Titcomb, which is more of a commensal in rats, and I know it's highly pathogenic in felids and for the veterinarians in the room, but we don't think it has serious pathogenicity in rats here. But we see a little bit of variation across this habitat gradient depending on the village, but again, maybe a generally declining relationship. And then the rat hookworm was sort of uniformly common across all of our distance away from the village here. It was also extremely prevalent, 75+% of rats had this hookworm. They had an array of other parasites, but these are just the 3 most common that I'm featuring here. Now let's look at our native small mammal here. So microgale also had coccidian parasites, and again, high prevalence, but very variable depending on what village it is. And one thing I want to draw your attention to is that, you can kind of see it, but not very well, I've sized these points based on the number of animals that we trapped in each of these trap grids. So you will notice that perhaps there's something going on in terms of how common that animal is related to the prevalence, right? So in our village S, if you remember, open landscape, not a whole lot of trees, we see fewer animals and also lower prevalence of their parasite. We also, interestingly, see low levels of rat hookworm. We don't know if these correspond to actual infections because this is metabarcoding data and not confirmed infections, but curious that potentially there's maybe something going on there, at least in terms of rat hookworm larvae being abundant in the landscape, and we're picking that up, whether it's a true infection or not, across those different landscape or different areas of the different distances coming away from the village. And then finally, what's I think most interesting is what's happening with this high prevalence mystery worm. There is a parasite that we found with pretty high, almost 100% prevalence, so very, very common in our more semi-intact areas here. But what's happening is as we get closer to the village, that worm is going away. And that is happening even though, as you'll see by the size of these dots, we still actually trap quite a few of the microgale there. Now, curiously, this parasite, when we sequenced it and then blasted it in GenBank to see what it was, the closest thing that showed up was a marsupial parasite called Vianea, which I had never heard of. And there are no marsupials in Madagascar. And even then it was like 80% similar. So what we're working with here is something that we don't even know what it is. So a next step here is to actually try to figure out what that parasite is. Answering that call about a global parasite conservation plan and maybe documenting this particular organism. So why is this happening? So the parasite ecologists, disease ecologists in the room are probably like, I know, density dependence. When you have more hosts, there's probably more transmission. And we definitely saw, as we move away from the village, we see an increasing number of native species that are trapped. So again, that's since our trapping effort was consistent across all of those areas, that's our proxy for the density of the animals. So again, more animals, more transmission, and then the opposite is happening with our invasive species. So that's definitely something that's going on related to that. But as I mentioned, we still see a lot of those microgale. So what Matan is doing is he's looking at all of these parasites that we found, and then he's looking at how each of these different covariates explain their distributions across the villages, and breaking it down by the proportion of variance explained by each of these covariates. So each parasite kind of has a profile of what best explains its distributions across different hosts and contexts. And then, next slide. Okay. Okay. Then Kayla Kaufman, who was a graduate student, now a postdoc at Stanford University. She is looking at what predicts multi-host parasite sharing. So we're seeing some of those parasites that we found in native species, crossing across the native and invasive species boundaries. And so she is mapping that probability of cross-species transmission. And what she's found is that forest edge distance, so this is really happening at those forest edges, is a significant driver of that. Okay, so our takeaways. We get opposing effects of human land use change on parasites in different species. Those effects seem to depend on the parasite characteristics. And we are really seeing this undescribed parasite diversity that is potentially going to be lost in those habitat, in areas where there's habitat degradation. So our 3 One Health lessons, zooming out. So in terms of blastocystis, we found that not all intuitive networks matter. So zoonotic infections, not important, and actually not friendship networks, but actually neighbor networks. Hookworms, different pathogens, even those that seem really similar and have similar transmission pathways, but just different host ranges, move through very different pathways, and ones that depend on the geography of the area that they're in. And then wildlife parasites, that disturbance reshapes disease ecology in opposing ways for different species. And so a simple rule that we get out of this is that ecology explains much of these patterns. So I'm a really strong advocate for any One Health work really thinking about ecology. But ecology is really big and it's not actually that simple. So our big takeaway is that One Health approaches that combine the expertise of different fields and that look at sort of create a menu of different transmission options that we can then rule out and figure out what's the most plausible transmission route is a really effective way of identifying which of those ecological aspects are relevant for transmission and thus intervention. And I just want to close by recognizing this is even just like a subset of all the people who are involved in these projects across the world, but a huge array of different folks who have contributed. Again, a huge amount of work, and I just talked about a tiny sliver of gastrointestinal parasites, but there's a lot more work going on in terms of antimicrobial resistance, viruses, and even human movement networks in terms of public transit as well. So a lot of cool stuff coming out from the team. And with that, I'm going to close, and I will I'll be happy to answer any of your questions. Also, feel free to email me at the address above here. And with that, thank you very much.
So we're going to bring the ice cream out for people in the room, and there's also swag. And then I'm going to monitor the chat, and if you have questions, just speak loudly because we're having audio issues. But thank you, Georgia. That was awesome.
Thank you.
Questions?
Yes.
Maybe I missed this part, but does your team include people from institution in Madagascar?
Yes, yes. So the institution called Association Vahatra, and one of our main PIs, Wangiswari Malala, she is one of our 2 central PIs there, and so they're based in Antananarivo, but we also involve the folks living in the villages in each of the 3 villages for this project, but all the others in coordinating, and we work with local public health officials. So they're the ones, while we can't make any recommendations in terms of human health work because we're, over here in the United States. What do we know? We share all of our information with the Malagasy Ministry of Health.
I mean researchers.
Sorry?
Researchers.
Yes, yes.
Researcher and governmental.
Yes, yes. Yeah, so I don't know if you saw one of our trapping, our head of trapping, he's a postdoc with Association Bahatra, and Association Bahatra also supports several PhD and master's students through this work as well, but they're mostly focused on the small mammal aspects.
All right, thank you.
Yeah.
Thank you for it.
Oh, no. Yeah, no, but it's a good and very important question. Yes. Yes.
If you said who's funding this large project, I didn't hear it.
Oh, I didn't. I just had the logo at the bottom. We're funded by both the NIH and the NSF, as well as several Duke collaboratory grants to Duke University in particular. Yes.
You mentioned with the blastocystis that there is this relationship with proximity to the river.
Yes.
How does that work?
Yes, good question in terms of the way it may be occurring is that blastocystis could be in the water sources, for example, and people who live close to the river are getting their water sources from the same areas of the river, and therefore they're exposed to the same subtypes, and they tend to have the same infection patterns.
So then the people who are further away from the river are getting their water elsewhere, or are they also going to the river?
Yeah, so they have established wells in the, I should mention that, in the village. So they're sort of also different, that's not the only water source, so there's also additional areas of landscape they could be getting water from.
And is that part of the testing that Christina and all are going to do, or look at the wells and the river?
Yes, she's gonna add water in rice fields, because also blastocystis can contaminate food. So people working in rice paddies, there may potentially be infection risk there, as well as the river. Yeah, it's a good question. Yeah.
Nice job, Rebecca. Your hookworm data, where you had co-infections, I think, made me wonder about your blastocystis infections.
Yeah.
And is there a possibility that you have co-infections there too, and so they harbor multiple subtypes, and only one is kind of growing out? Do you have any evidence to show that there are multiple infections in these animals?
In humans?
Humans,
yes. So thank you for asking that. We do have triple infections. We do have individuals who have blastocystis as well as hookworm. I only looked at this, very early, much earlier on in terms of any blastocystis infection rather than specific subtype infection, to see if that had a relationship to hookworm infection. But when I just look at blasto by itself, all of them together, there's not a pattern. Yes, there's an overlap, and it's random. However, obviously, based on this, we need to look at the subtype to see if that is what might be, or if there's any variation.
Yeah, because I'm wondering if you can completely rule out zoonosis if there's a subtype that is maintained, even if it's a minor subtype in some of the animals, and then once it enters a human, that subtype that grows better or lives better in the human becomes the predominant subtype.
Yeah, that's interesting to see if there's, one thing I guess, it's fine. I can show you later. I do have a map of the different, subtypes across village, for the different individuals, and then also intersecting with the hookworm infection patterns as well. That's an interesting hypothesis that we should test. Yeah.
I have a question. Did you get an idea of intensity of hookworm infection?
No, great question. So for those who may not know, gastrointestinal worms tend to vary enormously in terms of their intensity. Most people have low-level infections, and a few individuals have really high infections. Unfortunately, with metabarcoding data, we just can't reliably measure that at this point, especially when we're using a locus that captures lots of different things. The best way to test that is to do egg counts, or to use a really highly refined qPCR test that's designated for that specific worm species. And we took a more broad approach rather than that really tailored approach. But if we wanted to do that, these results would really inform, okay, we should do that if we want to look at intensity and how that maps across the different locations.
Yeah, I was just thinking about implications to health. I mean, you're probably aware in marine mammals, hookworm infections cause severe, like life-threatening anemias.
Yeah.
And so just wondering about, and so we were looking at intensity and then infection and seeing the impact in, emaciation, et cetera. So just wondering about burden and what that's doing in terms of health of animals and the dogs and the people.
Yeah, it would definitely help close the loop in terms of the health effects, because we see such high prevalence, but not a clear association with negative health impacts aside from the signal of metabolic stuff that's going on, but not serious metabolic issues. Definitely something that would be a really good follow-up.
What are you doing to identify the unknown parasite? Are you doing necropsies, parasitology, pathology? There's many ways to identify the parasites. So I wonder if you're actually doing actual necropsies. That would be very valuable.
No, not with obviously not with humans or domestic animals. So we use metabarcoding for that. And so we use the ITS2 gene, which is a pretty well established one for the nematodes in particular. For the small mammals, the Association Bahatra functions as sort of the collection center in Madagascar, so all of their specimens are located there. But it's sort of in the long-term plan to go out there to try to catalog some of the parasites, but they don't currently have a parasitologist out there, so that's like one of the long-term goals is to increase the parasite, ability of scientists to understand parasites in Madagascar, where the specimens are.
And I'm surprised you didn't find toxoplasma. Toxoplasma is more prevalent than blastocyst. So
we think there probably is toxo. It just may be that the particular primer that we use is better suited for blastocystis. And so we are following up on these data with a different set of primers that will hopefully, remove some potential biases. We don't know for sure. We also did not detect very much Giardia, but we think it's in the system. So we are going to go out again and try to see if there's gaps that we've missed here as well. But again, that's pros and cons of metabarcoding in that you can get a lot of things, but there's some biases and some taxa. Yeah. Yeah, and I saw a question in the back there.
One thing to keep in mind, I think that's just been studied in the field of virology, is persistent infections in which an animal can be persistently infected, excuse me, without expression, without replication. Sorry about that. I guess there's, persistent infections can be manifested by kind of lack of expression, lack of replication, cryptic infections that are still relevant. So for instance, in the case of blastocysts, likely you have exposure to different forms of the parasite. So in some cases, there may be actual infection without replication, reproduction, and then ability to detect that infection. But it can still be of importance in terms of immune responses, responses to infection by other parasites, other blastocyst parasites, etc. So there's a complexity that probably may exist there with any parasite, I don't think the phenomenon has been studied as well with parasites as with viruses, but could apply.
Yeah, that's really fascinating, and I mean, based on our results here, we would hypothesize that looking for subtype 5 would be, something to look at, again, because that's super high prevalence in pigs and seem to be one that humans would probably be more likely to come in contact with. But again, we didn't find it in any of the human stool samples at all. But they're surely being exposed because there's a good number of pigs and they've got 50% prevalence at least, like even with our detection methods. So yeah, that point's really well taken. Thank you.
Anyone online want to come off mute? Any more questions in the room?
Great question. Thank you.
All right, well, thank you all for coming again. Thank you for that.
Sorry, I'm not on the laptop, everyone.
And the link to the presentation will be eventually on the One Health Institute website if you guys would like to see any of that again. And for those of you in the room, please enjoy ice cream and each other. And we also have, oh my God. Swag. I was like, what's an S? My brain. And thank you all for coming. Appreciate you joining us.